Hello, everybody. I am really excited to welcome you all on behalf of US HUPO organization to the office hours. And I am really happy to have the opportunity to host an event like it is today, because of course, in the new online world, even though we can have various talks and lectures, what we're really missing is the equivalent of the hallway conversations with some of our friends and colleagues, or noisy bar conversations, where we can just say, hey, how are you doing this, right? So what worked for you? Oh, my experiment failed, so what happened? And this is something which is very difficult to reproduce in a kind of formal Zoom setting. So we came up with this idea of office hours, which approximate this to the best possible way, an informal discussion about things which happen in practice and some tricks or tips or just general experiences about particular topics. And so today I am super excited to host our two speakers, Lindsay Pino and Brian Shorrell, who are experts in many things, including data independent acquisition, both experimental and computational aspects of that. And Lindsay and Brian have a long history of working on educational events, and they have a long history of making important research contributions. In particular, both of them last year were named Rising Stars in Proteomics and Metabolomics by Journal of Proteome Research. So a couple of words about our speakers. So Lindsay has an undergraduate degree from University of Pennsylvania, after which, if I remember correctly, she went to Peace Corps in South Korea. Is that right?
Yeah, I did a Fulbright scholarship to South Korea.
Fulbright, yes, Fulbright in South Korea. But after that, she came back to academia, and so she worked for a while at the Broad Institute, working in proteomics, and then she did her PhD in the lab of Mike McCoss at the University of Washington, and then she moved to UPenn, where she was a postdoc, and currently, oops, sorry about that. Currently, she is in charge of a company that she co-founded with several other colleagues, Talus Bio, which works on using mass spectrometry-based tools for various aspects of biological discovery. And so Brian, on the other hand, so he has a undergraduate degree in chemistry from Reed College. And after that, he also had an interesting career path. He co-founded Proteome Software together with his colleagues, which produce cutting-edge data analysis software for proteomics, in particular Scaffold. You may have heard of that. And then eventually he went back to academia, and he earned his PhD also at University of Washington in the lab of Mike McCoss. Then he did a postdoc at the Institute of Systems Biology in Seattle. Actually, not a postdoc, but an independent fellowship at the Institute for Systems Biology in Seattle. And now he is a faculty at the Ohio State University. And so I'm really excited to listen to the conversation, to the informal conversation of these two experts. And I hope that this will be as interactive as possible, and that all of you will take an opportunity to ask questions either in chat or raise your hand, or, well, just speaking up. So you all can share, like have voice and share your cameras and even share screen if necessary. So without further ado, let me just welcome Lindsay and Brian, and I'm looking forward to hearing from them.
Awesome. Thanks for the fantastic introduction, Olga. I think just so that we're all on the same page, I thought I would show a couple of slides and then we would have a conversation. So I think that the majority of this should be open group so that we can ask questions and go back and forth. This, as Olga said, this should be like a hallway conversation. It should be a fun interactive experience. So I want to get through these slides pretty quickly. Okay, so DIA for DDA people is kind of what we were looking for. How do you set up a first experiment if all of you, for the majority of the type of work that you've done has been DDA or PRM or that type of experiment? So first, just so that we're on the same page, proteomics, we study proteomics by monitoring proteins that are generated by cells, and that these proteins we typically will digest into peptides by cutting them with digestion enzymes, and then we're looking for those particular peptides. Oftentimes the proteins we care about, so for example, like these green proteins or orange proteins, are hidden in a background of all of these other proteins presented by the cells that we're monitoring. So when we study these types of things with data-dependent acquisition using normal sort of mass spectrometry-based proteomics, we might be looking at peptides that are eluting with specific precursors at across a little retention time range. And so each of these sort of distributions represents kind of a peptide. And with DDA, we let the signal describe what we should be monitoring. So for example, we see high signal at this peak and we monitor 507 and we collect a fragment spectrum, and then we do the next for the next spectrum and the next spectrum and the next spectrum and the next spectrum. This is a great method for trying to dig very deeply into data sets where we let the data decide which peptides we should try to be sequencing at any given time point. But this is kind of like a backwards way of thinking about the problem that we're interested in, right? If we're interested in monitoring a handful of proteins, that might be analogous to like looking for a book in a library. And this is shamelessly stolen from a slide from Jarrett Egerton. But the idea basically is if you were going into the library looking for a book like Harry Potter book, you would like as the best strategy, you probably wouldn't take a bunch of books off the shelf, maybe the biggest books that you could find, and then pull them out into a cart and then go through them one at a time and say, I found Moby Dick, I found Pride and Prejudice, I found War and Peace. I didn't find Harry Potter, therefore, is it in the library? The answer to that is no, you don't know really. And so a better way to kind of think of this is that if you were to use a little bit of information about the book that you were interested in, so for example, the Harry Potter series were written by J.K. Rowling, you just go look in the R section, and then you would find whether Harry Potter was there or checked out from the library. And that seems like a much more logical way to approach a library question, but also a question for proteomics too. And so what happens when we don't see peptides like these peptides, this blue one, for example, that we didn't collect an MS2 for? Well, there's a couple of strategies that you can do for that. One of them is a targeted approach where you constantly collect fragment scans for that particular precursor. So you know what that mass for that precursor is. And so you constantly are collecting those scans all the way across time. And then you would compare sort of the fragmentation profiles for those fragment ions, which you would see as these sort of distributions because you're collecting them over time. You would compare them to synthetic data. So either predicted spectra or spectra from synthetic peptides that you've generated. And you can kind of think of this as like a mass spectrometry-based Western blot. So DIA is somewhat similar, but instead of looking at narrow precursor windows that you've selected for specific peptides, we're looking at wide precursor windows that are sort of tiled across the entire MZ range. And so there's a range from 500 to 525. And in that range, you'll co-fragment, say, these three peptides at the same time. And you'll monitor these and look at them as specific spectra. So there's kind of two ways to sort of think about interpreting this data. One of them is what we call spectrum-centric or maybe signal-centric interpretation of this sort of multiplexed DIA measurement, where you've got precursors signals that sort of related to fragment ion signals, and you say because they follow the same peak shape, they should be related, they should belong to particular peptides. Another way you can kind of think of this is what we call peptide-centric searching, where you have a peptide of interest, maybe one of those ones that you were interested in before, and you look for, is there any signal that sort of represents this peptide in my system? So another way to think of this is also, so this would be sort of standard DIA analysis. And then a final way to kind of think about this would be looking at what we call gas phase fractionated DIA analysis, where instead of collecting one set of windows, you collect a bunch of set of windows for the same sample. So you inject that sample multiple times with narrower windows. So instead of looking for 25 MZ wide windows for across your entire MZ space, you might look at 5 injections of 5 MZ wide windows. And at a certain point, you end up collecting data that is essentially like PRM data, where instead of monitoring a specific peptide, you're monitoring a range, but that range is quite narrow. And therefore, you're probably only going to get one peptide at a time. And then you can go back and mine that data for other types of peptides at the same time. So that's kind of like the background for the DIA style methods, not just from and it sort of thinks of DIA as sort of an extension of PRM rather than an extension of DDA, because I really do think of it as PRM is essentially a DIA style measurement, because you're sampling very regularly at specific time points that you pre-configured. So with that, we had a couple of questions that we wanted to kick off, just sort of having a conversation about, so questions like, why would you bother with DIA over these other types of methods? But then also, when are these other types of methods maybe good enough for doing analysis? Finally, how would you set up a method like this? And then sort of what are the challenges and pitfalls for doing this? So these were kind of the questions that we wanted to ask each other, but we are super excited about having your questions here and trying to talk about all of the different things that you've run into when trying to analyze DIA, as well as what has sort of stopped you or gotten you sort of out of your groove for being able to collect DIA data, and what can we kind of do to help you get back into that and figure out what's going on. So with that, I'll stop the share so that we can sort of just have this talk. So Lindsay, let's start off with you. So why would you bother in these cases with DIA versus DDA or PRM?
Yeah, so for most of the experiments that I'm doing, I'm interested in quantitative comparisons, right? So I want to do some sort of differential abundance analysis. So DIA typically is going to be better for those kind of experiments if quantitative comparison or a differential comparison is the experimental question. DIA is fantastic for that. I also, especially at Talus, we're really interested in like very large numbers of samples, right? Like hundreds to thousands of samples. So another good opportunity where DIA will scale with you. So those are kind of the two main use cases that have drawn me to DIA. I'm curious, anybody else, or Brian, if you've started experimenting with DIA, what are kind of like the specific use cases that you have found?
Yeah, and for everyone, just please jump in, turn your video
on.
Yeah,
for sure.
And
this is very
questions.
Casual.
Yeah, or if you're not comfortable with the video, that's totally fine too, and just enter in something in the chat or whatever. That's also good too. We'll keep an eye on that. So just to kind of think about those types of questions, I think that DIA can be really good at certain types of high dynamic range problems as well. So for example, if you're analyzing serum, I think it's a little bit easier in some ways to analyze that data with DIA, because with DDA, the instrument just is constantly sampling the same types of proteins over and over again. It's either like albumin or hemoglobin or something like that. And that with DIA, at least you're actively telling the instrument to like think about other ranges of peptides. I guess another circumstance along those lines is that oftentimes it can be good for doing needle in a haystack problems. So like if you're trying to find something that's pretty rare. So Lisa has a question. Waldron? Yeah.
Yeah, hi there. So I've just recently started with DIA, and I'm not sure if this should be really obvious or not, but how in practical terms do you set up your DIA library using the gas phase fractionated runs and IRT peptides?
Yeah, that's a great question, because IRT peptides definitely don't come out in every fraction either. So that's I think a really excellent an excellent question. So what's going on here is when you're doing gas phase fractionation, you're doing a bunch of injections of the same sample over and over again, and you're sampling specific ranges. So for example, from 400 to 500 MZ, and so that's the entire experiment. And then from 500 to 600, that's the entire experiment. And so some of the peptides in IRT peptides will elute from 500 to 600, but not all of them. Very few of them in the end will. And definitely not enough to kind of figure out retention time alignment for them. I don't know about you, Lindsay, but I have actually stopped using IRT peptides or retention time standard peptides of any kind. Like I'll use peptides standard peptides in my samples just to make sure that I am injecting the right amount, or the amount that I expect to be seeing on column is actually the right amount, but I don't use them for alignment in the end. And the reason why is because oftentimes you have, I mean, these types of samples, you have hundreds of peptides, thousands of peptides in those samples that you can align from run to run. That aren't IRT peptides, they're just endogenous peptides. And so if you know a little bit about them ahead of time, you could just use those for alignment instead. And so that's how, like, we treat this problem in Encyclopedia. We actually don't even use IRT peptides at all in our database searching or library searching. And we just sort of scan for all peptides, and then we use retention time after the fact. As a way to kind of do that analysis. But I think that either thinking about your endogenous peptides as retention time standards, or forgoing retention time standards at all, I think both of those are good ways to think about it. So what do you think about that, Lindsay?
Well, I was going to say, what do you think about that, Birgit?
Yes, so thank you, Lindsay. Yeah, I'm an extremist, I would say. I mean, we have totally stopped using DDA altogether. I mean, we use DIA now for everything. And I mean, we do still add IRT peptides in, but algorithms nowadays don't really need them anymore. But I mean, even recently we did some high pH fractionation to build deep libraries. Even that we did in DIA. So what we do is actually we kind of do a similar thing to what you do, build the libraries directly from the DIA files, right? And so we use Spectronaut for that. But then, yeah, there's really no need for us to do DDA. I mean, I think in the early days, DIA was a little bit behind with the identifications, but the algorithms got so much better. So yeah, I mean, I think I love DIA.
Yeah, Noah.
Hey, can you recommend kind of a DIA for dummies? Like, having never done one experiment, you know, what would be the lowest? And to me, and doing anything new, there's always, like, you know, the activation energy of, you know, well, what are the settings on my instrument? What software should I use? Which software will I download and actually be able to make run without having to jump through more hoops, which would make me put it back on the side. So, you know, what's the easiest route to just get an experiment done and get a feel for how it's going to go and what it looks like?
Lindsay, you want to do this one?
Yeah, I will be honest, I don't think there really is a good, short, succinct, like, paper that'll tell you to do that. They're a little bit lengthy. Birgit.
I mean, your paper, right? The one in MCP, I think, is not a bad one.
It's not a bad one. It's thermo-specific. It doesn't really give you kind of like the breadth. It's very deep, if that makes sense. It's a very deep paper. You can kind of skip to the end, pay attention to the tables that have instrument settings in them. If you have a thermo instrument, it has all the instrument settings, and it has a guide for how to use Brian's Encyclopedia software to analyze, which is free.
Lindsay,
you
could
can
you
post the
link
URL
that
for
paper?
Yeah, I will link that paper now. And Birgit, if you wanted to jump off of that, you have a paper rec?
Yeah, so so Noah, that's a great question. And we've been doing DIA on triple TOF SciEx instruments for over 10 years, right? So we had our methods totally in place. And then we got a thermo instrument in Eclipse, and basically we had the same thing like you. We were all of a sudden new users, right? And had to find out what should I use. And we tested like 3-4 different settings from different papers. And what we really love and now do on the both the Explorers for AD and the Eclipse is a paper from Roland Bruderer, and he had a good method there. He's from Diagnosis, and so that's what we've been using since. But you're right. I mean, this is a challenge, right? There's so many different people with different settings, right? And yeah, I'll see that I link this paper in. Yep. Mm-hmm.
I think for me as a DDA user, I'm a little overwhelmed by the idea of the libraries, whether it's do these libraries come from empirical data that I've collected? And then my question is always, well, how do I know my library is good enough? Or
the beautiful thing with DDA, with DIA these days, you don't even need a library. I think a lot of us are actually moving away from even using libraries, and instead, a lot of these tools and approaches, you can just download these predicted libraries straight off the internet and plug them into your software of choice, whatever you happen to be using. So you don't even have to worry about libraries anymore. You can just do your DIA and analyze it straight without doing these kind of fractionations or what kind of library to build.
So along those lines, there's a source that I put into the oops, sorry, that was the wrong place for that message. There we go. So there's a good source for pre-predicted libraries over at Proteomics DB for PROSIT. PROSIT is sort of one of the tools that people have been using for generating these libraries. So there are some libraries that you could just download, but you can also just use the website for making your own type of libraries as well. So that might be a good place to get started. You can also, there's different sources. I know that on the encyclopedia website, we have the pan-human library. If you're working in human stuff, that's great for starting out. That's from the Aebersold lab. Another good source is if you're doing phosphoproteomics, we have another phosphoproteomic specific library, for example.
Oh, can I say something?
Yeah,
yes.
definitely.
Oh, yeah. I mean, this is also a fundamental question. And for years, for example, for human, I have used the pan-human library, which I think is great. And there's actually quite a lot more libraries that have been coming out now that also were made from different tissues. However, we also have a lot of projects that are very specific, right? There's like some mouse cortex or mouse striatum or PTM, as Brian said. And for that, there's really no, or like we work with C. elegans, so there's not such great public libraries out there. So we have just started really making our own libraries. And when you use this approach where you build the library from the DIA files directly, right? I mean, per se, it is a library that is sample specific. So yeah, I think that's what we know. I have kind of shifted a little bit from previous using more these DDA built libraries or big libraries that were available to now often use sample specific libraries.
Mm-hmm, mm-hmm. Yeah, I think it's definitely a very useful strategy to do sample specific libraries. I think it's best to do sample specific libraries when they're not when they're built off of these gas phase fractionated approaches. I think the ones that Lindsay describes in her paper, I think is a great way to build libraries very inexpensively without a lot of effort, because you really just need like 6 injections of PRM quality data and then analyze that using, and you can, this is like shooting fish in a bucket. It's very easy to identify peptides when your quality is better than DDA to be able to detect all of the peptides in those samples. So I think it's a really good way to go. Just sort of looking at the chat, Nico asks, have we tried to use DIA with metaproteomics experiments? This is a great question. I actually have started to do some metaproteomics with them. I think it's an interesting challenge because libraries become much larger when you start to look at multiple species, and there you have to start thinking more about going back to the concepts of DDA-based libraries, actually, to kind of narrow down your search space, because thinking about every microbe in the ocean, for example, can be a challenging problem, but it's definitely something that we're excited about doing, and I think that there's a lot of opportunity there because oftentimes you need to monitor things that sort of drop out in one microbiome versus another microbiome, and you need like this ability to be able to call a 0 point for the quantitative analysis.
I guess along those same lines of alternative uses for DIA besides proteomics, there was another question from Fabiola saying, could you use DIA for other types of compounds like flavonoids? I certainly have not tried DIA with anything other than proteomics. Anybody?
Yes, so in fact, there is a group in Oregon, no, in Corvallis, Oregon, and they have a triple top 5600, and they use this exclusively for proteomics. And I have to think of the name, Fred. I will find the. I mean, I'm sure they have, and you know, like some really nice papers out for that. But yes, I think totally, absolutely. And the question is then, yeah, what do you have to build your own libraries? But maybe in these papers from this Corvallis group, I can forward to you. And I just wanted to say, I did post in one paper that we had in Methods Molecular Biology together with SciEx. This is a step-by-step, how do you set up a DIA on a 60 or 6600, right? Yeah, and the other paper I will still find.
So Lindsay in the chat, Kinbo asks, how do you set up dynamic windows, so the isolation windows for dynamically assigned windows, or variable width windows, I think is how they're normally called. And on what circumstances is it smart to set up variable width windows versus sort of fixed windows of specific or circumstances? I think that there's choices there in lots of cases.
Yeah, it's an interesting question because it kind of gets into the question of, I think for a lot of DDA applications, you can just kind of apply the same DDA method no matter what your sample is. Mm-hmm. But with DIA, you have to think a little bit more about what the types of samples are. So as far as a variable width window, if you have some type of sample in which your peptides are not distributed evenly across the MZ range, that would maybe be a good opportunity to use these variable width windows. As a disclaimer, I myself don't use variable width windows for anything that I do, but I'm also usually doing some like complex sample where I can expect the distribution of mass to charge values to be more or less even across a range of precursor values. I guess I'm kind of curious, what are the kinds of applications that you were thinking of using for variable width windows?
Actually, I'm using it for to test the samples with the high abundance of albumin, but it's actually for some mycobacterial samples. So in that way, it has a lot of like a high abundant protein that we don't interested, but we really want to see if we can use variable windows to just remove those interference.
Yeah. So I think that this is a really great point. So the variable width windows, I think, is a really cool technology to be able to say some windows have a lot fewer peptides and you shouldn't need to spend as much time monitoring them. I think that that approach works really well on certain types of instruments, but I don't know that it's the best approach for all instruments. So for example, by using variable width windows, that also means that you can't use other types of technologies like staggered windows, where you shift windows over every other cycle to get twice as much precursor isolation for your peptides. So that strategy in our hands on Orbitraps performs better than variable width windows. But that has to do with the fact that Orbitraps scan fairly slowly. And if you're using a TOF, I think it makes more sense to think about variable width windows. That said, I think that every sample type needs new variable width windows. We've looked at like phospho, for example, which shifts the mass ranges completely for what window sections you would actually want to set up. So I think that you can use variable width windows, but you shouldn't just use them blindly, assuming that they work for every circumstance. The other aspect of that is I think that oftentimes if your windows get very wide, you can have co-eluting peptides that have the same, have many of the same fragment ions, but not quite the same precursor mass. So for example, a peptide and an oxidized form of that peptide will elute within a few minutes of each other and share a lot of the same fragment ions. And if your precursor isolation windows are wider than say 16, or in many cases 4 or even 8, then then you end up not being able to tell the difference between oxidized peptides and unoxidized peptides. The same thing happens for sequence variants or other types of peptides that you would potentially see in the same isolation window. So I think that's a really good question. Does that kind of answer? You have to ask that question from both sides. Like, there's a benefit for a variable with windows, but there's also a negative in that you can have some really wide windows too.
Yes, thank you. Yeah, I agree with you.
In my experience though, in SAS instrument, it does improve a little bit, but triple tap instruments are so fast that the improvement is, it's not that significant. I think it improved like 5% or something very minimal. And if you are using a SciX instrument, which is kind of where the variable window will be more beneficial, you can email them and request the Excel spreadsheet that they have to calculate those windows. Otherwise, there is an RR package, but it's not supported anymore. So, yeah.
Yes, I wanted to say though, we used variable windows also on the thermo instruments. So, I mean, I think variable windows has a value, right? Because there are certainly areas that are super busy. So there it's really beneficial to have a bit smaller windows versus say at the end, right? Like the high end is a lot less, busy. So there we have bigger windows. Mm-hmm.
Yeah, I think there are choices that you can make, and the choices depend on the specific experiment that you want. Camille, since you have your hand up, did you have something you wanted to ask?
No, sorry, I just wanted to interject about the variable.
Perfect. Okay, great. Awesome.
So I have something. So at the moment in our lab, we mainly do, we're starting to do DDA. The problem with our lab is that we don't have a great facility to generate libraries to fractionate peptides so we are mainly relying on softwares like DDA that does library free searches. My main question is, what I notice is decreasing the window width. So at first I tried 25 MZ window, that worked fine. And then I reduced the window width, and that doesn't give me greater number of peptides. So what's the advantage there? Like, is it because of the machine, or is it because of some other thing? So what I have noticed is that the increasing number of windows doesn't increase the peptide number identifications.
So I'm not sure I got all of what you were saying. So you were breaking up a bit. So if I'm not answering the question or restating your question, then enter it into the chat and we'll try to go from there. But I think what you're saying is that if you add more windows, smaller windows, you should get more peptides, but sometimes you don't necessarily. Is that right?
Yes, I don't see an increase in the number of peptides identified compared to a 25 MZ window method to a 64 window method. So 11 by Z, so 32 windows, 64 windows. So increasing the windows doesn't really increase peptide identifications.
So, Mathran, I think this is a great teaching idea. So what happens when you increase the number of windows? You obviously have to spend less time on each window, right? You can park the instrument on a specific window and collect ions for a certain amount of time. And when you start talking about 64 windows or 120 windows, you end up having to spend less and less time on that specific time point. So for example, when you go to 120 windows, like you are at best trying to analyze and accumulate ions and then make a measurement. All of that has to happen within 20 milliseconds or something like that. And it's oftentimes just not enough time to be able to make a good measurement. And so oftentimes we find that if your sample is very complex, if you've got a lot of peptides at any given time point, then you can start to shrink your window size down and collect fewer or spend less time on those windows. but that in sparser samples, you actually need to park the instrument on those ranges for longer to be able to get good enough signal to be able to analyze and detect peptides out of those windows. Otherwise, the signal is just not good enough. And this also depends on the sensitivity of your instrument. So for example, it's possible, for example, to run an Orbitrap, like an older style Orbitrap, a QEHF, for example, at 12 MZ wide windows, such that you could do 20 hertz, so 20 scans per second. So you can do that, but the quality of the data that you get out is pretty poor. But so, for example, if you used a newer instrument, you can actually scan that fast and get reasonable type of peptides or signals for those 20 hertz scans, but only in certain types of samples. So that I think works really well in very complex samples, but oftentimes we still want to go back to that kind of like 24 MZ wide windows in sparser samples. So if we're analyzing like serum or something like that, I think where there's fewer types of proteins or the complexity of it is a little bit less, or the dynamic range is a little bit more, then I think that oftentimes in those circumstances, you actually need wider windows and to spend more time accumulating ions and accumulating signal to be able to analyze them. So hopefully that answers your question. If not, then stick it in the chat.
Yeah, I think this actually leads into another question that we had in the chat regarding the dynamic range. So like what would be kind of the dynamic range comparison of DIA versus DDA versus PRM for protein ID? So I usually never think of dynamic range in terms of identification. So I'm not entirely sure what that means. Do you just mean detection of low abundance proteins, Sean?
Yes, that's my question. In certain situations, we would like to identify those low abundance proteins, right? Would I think if extra benefit for this, DIA approach? Thank you.
Yeah, so I think DIA will absolutely be better than DDA for low abundance proteins, right? For DDA, to get those low abundance proteins, you're either going to have to do some complex depletions to remove the high abundance proteins, or you're going to have to deeply fractionate your samples so that you can overcome the stochasticity of DDA. DIA doesn't have that stochasticity. So when you have these, I think Brian described them needle in a haystack problems, where you have a very low abundant protein that you're trying to detect and quantify. DIA is actually probably going to do a better job of getting that protein ID for you than DIA would.
Yeah, I think that's a really good point. I think that the idea here is that if you can think of it like the Hubble telescope, right? Where if you kind of like point the telescope into the black regions of space for long enough, you can actually see galaxies and stars and all sorts of crazy stuff out there. And I think that that's essentially what we're doing with DIA, because we're programming the instrument to look in a window that you don't see a precursor for. But still try to collect fragment ion spectra for that range and to sit and park on that for 50 milliseconds or something like that, to inject ions for that long and say, well, show me what you see there anyway, even though you don't see a precursor there. And oftentimes we see peptides, a significant number, a surprising number of peptides. We, on Orbitrap instruments at least, we don't actually see precursors for if you're doing a full range scan, because you're accumulating, maybe 1 million ions for that full range scan to be able to stick in the Orbitrap and make a measurement. Your particular peptide might not fit within that 1 million ions or 1 million charges. But if you then narrow it down to this specific precursor MZ range, you can accumulate peptides or ions for those peptides, specifically in these ranges of the proteome where it's really complex and there's lots of stuff going on at once. Your particular peptide of interest might actually just be missed because it doesn't have enough signal to fit within that Orbitrap size, that 1 million charges. So that's a great question. Leanne, you have your hand up.
Yeah, so kind of along that discussion, this is also a bit of a naive question. Has anyone tried multiplexing, like TMT DIA, Peter is waving his hands. Have you tried, Peter? Because, you know, some of the benefits of TMT is the large sample size, which I think Edu is asking about, you know, replicates. And then also, you know, for modified peptides, which are in really low abundance and hard to identify, maybe DIA would help. So are people combining those technologies?
That one's a
Leanne,
hard
I'm
one.
just going to jump in
Yeah.
before Brian and Lindsay
Do
take
it.
this off. Leanne, you took my question. We're on the same
Sorry.
wavelength. That's exactly it. Lindsay, Brian, you guys asked at the start of this, what were the barriers for people? I've worked in a couple of different core facilities all over the place, and we have people that love their SILAC, their TMT, and sometimes convincing them to go label free takes a little bit of effort. So my question was going to be like Leanne, has anybody ventured in, or is it even worse? Or yeah, naively, is it even possible to do labeled DIA approaches? And I haven't done any, just to be clear.
So yeah, so before Lindsay talks about SILAC, which you should talk about SILAC, which is definitely possible. TMT is not really possible, and the reason why is because TMT actively puts all of the quantitative information in fragment ions that fall off the peptide. So you get good signal for all the fragment ions that represent the sort of sequence of that peptide, but then all of your fragment ions that represent the quant are jumbled together. So they're combined because it's all, you know, for iTrack, it's 114, 115, 116, 117. They're all the same fragment ions for all of your peptides. So all of that signal is repeated on top of each other. So TMT and iTrack, the normal TMT and iTrack don't work. You can use a complementary TMT and iTrack. That's where the mass falls off and you change the masses of the fragment ions that you're left with. Those do work theoretically, but oftentimes the amount of signal you get from them is so poor because of the efficiency of those fragmentation events is just not very good. That it's, having looked at some of that data, it seems like a giant mess to try to deal with. It is, I think, theoretically possible to at least use that technique, but you can't use a normal sort of TMT or iTrack technique. But Lindsay, you should talk about SILAC, and I'd love to hear you rant about TMT generally.
Yeah, so I think the question kind of boils down to what is the experimental reason these collaborators want to do labeled quant? I think the top reason most collaborators have told me they want to do TMT or iTrack is because it multiplexes. But does it really? Like, you're still doing all your individual sample preps, and then you're introducing an extra sample prep variation layer of doing the chemical labeling and then mixing them together before you run the mass spectrometer. So whether you're doing labeled or label free, you are really not saving yourself any experimental variance except for the instrument, which is the lowest amount of variance you're introducing to your samples. The instrument is not where your variance is coming in. It's coming in from your sample prep. So TMT and iTrack aren't saving you from that.
But don't you save yourself instrument time?
Yeah, you usually don't even end up saving yourself instrument time because a lot of times you're fractionating it out or running a longer gradient, right? Like, you might multiplex all your TMT samples together, but how many people multiplex their TMT and then run a 90-minute gradient, a single 90-minute gradient? Nobody, right? Like, so it ends up not really saving you any sample prep. It doesn't save you instrument time typically. And the variance that you are normalizing out of your data is not the largest source of variation you're going to encounter. So you're adding extra expense to buy that. You're adding extra sample prep time to do the chemical labeling. And typically there's like some extra runs in there to make sure your labeling efficiency and your mixing is going to be 1-to-1-to-1. So I usually tell collaborators, sure, like, definitely we can do that, but here's all the pitfalls. Here's all the reasons you might not want to do that, and do label free instead.
I should point out that the statistical analysis is also more complicated.
Yes.
Yep, I was just about to bring up Olga. Olga's group just did a comparison, maybe it was 2 years ago now, comparing TMT and DIA. And if you are super experienced in TMT, you get the same results as if you are experienced and comfortable with DIA. So you're really, in a head-to-head comparison, they're about equivalent. So you're not getting better data by doing TMT, is kind of the take-home message. So SciLac is kind of nice because you're mixing your samples together upstream of sample prep, right? So in that situation, if people really strongly feel like they are not good at prepping samples, maybe that's a good thing for them to do. And I would encourage them to prep their samples together in SciLac because they apparently have such large variation from their sample prep that they're concerned about it. There are some applications where that might actually be the case, right? Like maybe you're doing SciLac because you're going to do an enrichment and you want to mix your lysates together so that when you enrich, you're controlling for that. And I will say that's kind of one of the situations I'm still using DDA for rather than DIA. A lot of collaborators coming into the group and they are doing IPMS, they're doing affinity purifications, IPs. In those situations, it's kind of like it shouldn't be such a complex sample. It should be a pretty simple sample. So just giving them the list of what proteins were in their sample, I could just do a DDA for that. SciLac DIA, however, I have a whole paper on this that I'll toss in the chat. SciLac DIA for turnover, fantastic. I know Birgit's done it. SciLac DIA for turnover, fantastic. Outperforms SciLac DDA for turnover. So highly recommend you just slap your SciLac samples into your normal DIA workflow. And with a couple small tweaks, depending on the software you're using, you'll get much better turnover data. So a couple more hands up. And we got about
so Amanda, do you want to start?
Yes, great. Thank you guys so much for doing this. This has been really helpful. So I really like PTMs, and I also use a species that nobody else uses. So, I mean, not nobody, but there's not a way for me to just download a spectral library somebody else has already created. So is it impossible for me to look for PTMs with DIA using without doing a very sample heavy large scale fractionation study first?
That's a great question. So I guess the question I would start is to ask you another question in return, which is sort of what PTM, because I think that different types of PTMs require you to think about different types of things.
So I study cysteine oxidation. Okay.
Yeah, so that actually I think will be a little bit more challenging because it will change fragmentation patterns and that sort of thing. So in those cases, I would typically want to do, this is like a good case study where you would want to do some fractionated DDA ahead of time to be able to identify a bunch of those types of peptides to give yourself a leg up in trying to identify them in your DIA runs. You don't have to do anything different with the DIA, but I think starting with a library for those types of samples is a good thing. And oftentimes for modifications where you haven't seen it, even if, if a modification changes sort of the fragmentation patterns and massively changes retention time, I think you kind of do want to think about collecting a DDA-based library, even if that type of sample has been well analyzed, as long as there isn't already a really good library. So for example, for phospholipeptides in humans, we have a good library already for that, the Phosphopedia library. But if you're looking at phospholipeptides in mice, that doesn't really get you much. So I think that would be the first characteristic. I think as prediction engines get better and better, I think then that becomes a possible strategy to start just making predicted libraries for them, the same way that we do predicted libraries for normal gas phase fractionated DIA analysis. So like for building libraries using gas phase fractionated systems. I think that those would work pretty well. Honestly, the sort of difference of my organism is unusual is only an issue for that, like if someone has collected a library for you already ahead of time. Oftentimes those libraries it's not a bad idea to collect those libraries on your own anyway, in those cases though, for PTMs. It's good to kind of know exactly what you're working with in those systems. Mm-hmm. It's a great question though.
We just did succinylation on a monkey sample, right? Non-human primate. And there's nothing out there, not even really much for the proteome, and then nothing for succinylation. But yes, we did use these prediction algorithms, as Brian said, as part of SpectroNow Direct DIA to build from the DIA, build the library on the fly and then quantify. So yeah, you can do it, Amanda.
Nilanjan, do you have your hand up?
Yeah, I had a question. So I've started doing the DIA in a very common organism, the E. coli. And one thing that we found out that the small molecular weight proteins in the entire proteome of the E. coli, we could not detect them very well. Any ideas why this might be happening? Like, is it general or should we be looking into any particular direction?
Small molecular weight proteins or small molecular weight peptides?
Yeah, so
small
at
MC
the end,
peptides.
Yeah. So at the end, we just wanted to see which are the proteins that we looked from our data. Like from the peptide data that we got, we created something to see the proteins that we're looking into. And there we found that the proteins which are having the less molecular weight, those we are not detecting very efficiently. Very little of them are being detected. So that's something I'm looking for.
Yeah, I mean, I think probably a big part of that is if you have small proteins, then you have fewer peptides that can be generated from them, and you might not get a good tryptic peptide for those two particular types of proteins. But Lindsay, do you want to comment on that one?
Sorry, I was manning the chat.
Thanks. Okay, great. Yeah. So why might it be hard to find small proteins in DIA in those cases? I think that logically, like small proteins produce fewer peptides that are going to be likely to be good. That's probably the most likely scenario for what's going on. There's always a possibility that there's that you have small proteins that are being lost through the sample processing aspect to your workflow, but I think that there shouldn't be a reason why DIA should be worse than DDA for small proteins. For small peptides, there might be some reasons you could actually make that claim, though.
So one more thing that I want to ask you here, just because I'm starting and it might be a bit naive. So when we started different set of libraries, like we did some in-gel digestion, we did some in-gas digestions, and we wanted to find out where we could have gone different. But my question here is, how do I make a choice exactly? Like if I'm doing a general experiment without any PTMs whatsoever, how do I decide whether I should go for a DIA direct, like in Spectronaut, or I should use a in-gas fractionation or in-gel fractionation? What's your what are the main parameters that I should be looking for?
So I definitely think that it's a good idea. So if you've got a single sample and you're trying to analyze it and try to identify as many proteins as possible, if you have the ability to collect gas phase fractionated DIA, I think it's better than single injection DIA. Even if you're using a tool like direct DIA from Spectronaut, I think it's just easier to identify peptides. And so I guess you should think of these two as separate questions, right? So should I use gas phase fractionation or not? And should I use direct DIA strategies or not? And I think that direct DIA strategies work well. They work best with gas phase fractionation. So if you're doing like the PROSIT predictions and matches against those, or direct DIA and Spectronaut, or with DIA-NN, all of those approaches I think work better if you have gas phase fractionated DIA, because the samples the signal is just a lot less complex. So normally we do, like, if you've got a single sample and you have enough time to analyze it multiple times, multiple injections, then I think that's a good strategy to go. Or if you're trying to build a library, I think that's a good way to go for those types of experiments. And then you can go take your single injection data and look up those peptides using those measurements you made in the gas phase fractionated samples.
Just to interrupt for a second. So we have roughly 5 minutes left before the end of the call. We do not have to finish this, so this is a little bit up to our hosts, Brian and Lindsay. So let us know like what are the time constraints, but we can finish in 5 minutes if all things being the same, or give a little bit.
So Lindsay, what's your timeline like? Do you have another meeting after, or?
I do have another meeting after, so I will have to go, but we've got some threads on Twitter. Feel free to jump onto Twitter and ask any of these questions. We can continue conversation.
it is actually a really good idea to continue this on Twitter, because this is open to everyone and it has been very useful in the past. And I think Birgit has an announcement to make as well.
This is very timely. In fact, we had an executive committee meeting earlier in the year, and we will build a new DIA group, informal, just like this, but also applications, clinical evaluations, and just anything to do with DIA. And we will be able to follow and continue these discussions. So I think US HUPO will establish this group. Maybe we meet once a month, informal, right, Olga? That would be cool, right?
Absolutely. Absolutely. I think, and I can just read the feedback in the chat. Many people say that this is very useful. And I personally find that this type of conversations help me much more than just listening to a lecture. And so I hope that we will continue on Twitter, continue within this US HUPO framework. And so if you have more questions, so I would suggest that given that Vincent needs to go and we kind of capped it to 1 p.m. Eastern, we'll stop here, but continue on Twitter and maybe save questions for the DIA discussion group.
I think that's a great plan. Also, feel free to email any of us. I think that that's another good way to get access to us too.
Perfect. Thank you very much for participating. We had a very large group today, which is very, very exciting. And thank you again, Brian and Lindsay, for the very useful conversation.
All right. Thanks, everyone.
Yeah, it was so good to see everyone too. Thanks for tuning in.
Brian and Lindsay. That's right.